Thank you, Songli. So our first speaker for this morning is Dr. Ignacio Munoz-Sanjuan, and he is the CEO and chairman of Rumi Scientific with over 2 decades of experience advancing therapies for neurodegenerative diseases, especially Huntington's disease. At the CHDI Foundation, he led translational efforts spanning small molecules, ASOs, gene therapies, and the first PET imaging ligands for mutant Huntington. He also founded the nonprofit Factor H to support HD families in Latin America and was recently honored with the 2024 Public Service Award from the International Movement Disorder Society. Welcome, Ignacio.
Thank you. Thank you for having me. Let me know if you can see the slides.
Yes.
Okay, that's great. Well, it's a great pleasure to be here today, and I wanted to thank PBSS and particularly Son Lee, Adrienne, and Swati for inviting me and facilitating this symposium. My talk is gonna be focused today on some of the work that we are undertaking at Rumi Scientific on disease modeling and drug discovery applications using human organoids. Okay, all right. What you should expect to hear today is one, about the technology itself and why we find that it's useful in the context of drug discovery. And I chose an example of one of our leading programs targeted to Huntington's disease. So I will introduce some of the key concepts about Huntington's disease today before talking about a novel target that we've identified using the organoid platform BRD9 as a novel therapeutics for the treatment of Huntington's disease. Organoids are fancy new systems in use today in very different therapeutic areas. At Rumi, we're mostly working on CNS and kidney organoid applications. The first breakthrough really is derived from the ability to use stem cells, human stem cells that are either derived from the embryos, these are called human stem cells, or induced pluripotent stem cells that are derived typically from skin fibroblasts that are reprogrammed to become stem cells and therefore be able to generate the entire repertoire of cells in the human organism. The ability to use stem cells opens up a vast array of technologies for genetically engineering these cells to enable different drug discovery applications, and it provides the starting system for supporting drug discovery along many different aspects of the traditional development process. The second breakthrough is really scalability, and it's the ability of using organoid platforms, and in particular, our platform, which is a micropattern organoid technology, to be able to scale up the use of organoids to support large-scale chemical and genomic approaches for target identification and drug discovery support. And finally, the ability to generate a large array of identical human mini tissues, whether they're brain, kidney, lung, or the neuromuscular junction, enables deep analytical pipelines to understand the impact of disease or potential therapeutics on key mechanisms that may be altered due to mutations. So all three capabilities are necessary in order for the organoids to impact our understanding of disease modeling and support therapeutic applications. The micropattern organoid platform is our proprietary approach, which is to be able to grow tissues in 3 dimensions in a standardized reproducible manner that enables the analytics to support drug discovery campaigns. And I just wanted to take a couple of steps here. Typically, organoids are generated from pluripotent stem cells that are coaxed into becoming cells of a particular tissue, and therefore be able to model different aspects of the physiology of this tissue in vitro. For us, the organoids are also a unique experimental system that is complex. It contains multiple cell types that are physiologically connected to one another and enable the identification of mechanisms that may be relevant to the human condition. There's been a lot of differences that have been reported, not just in the context of the CNS, also in the immune system, and at a molecular level, where a lot of the genes that are associated as causative agents in disease or modifying disease progression in various indications require human-specific sequences that are not typically modeled in non-human systems. The second aspect is that these systems are incredibly sensitive to perturbations, enabling the ability to model cellular consequences of disease-causing or modifying genes in a way that enables quantitative understanding of how to modulate the primary function of these genes, something that has been largely limited in the context of neurodegeneration, particularly. The technology is also compatible due to its high throughput with chemogenomic, CRISPR, and nucleic acid screening, really enabling the development of multiple modalities as therapeutic strategies. The types of cellular mechanisms that can be modeled with organoids is really pretty limitless, and therefore enables an understanding and identification of quantitative phenotypes in areas that are typically not normally investigated in more traditional 2D systems or with heterologous cell lines, which is usually the starting point after a target has been identified. In the context of Rumi, we've applied this micropattern organoid technology to the understanding of lung mini tissues, particularly in the context of viral infections like COVID, in kidney and in brain. Within the brain or within any of these organoid platforms, the ability to modulate the fate specification of pluripotent cells becoming more restricted to different cell fates and different regions of a particular tissue also enables asking very specific questions about the impact of disease mutations on very specific cellular processes based on our understanding of the evolution of these disorders. So, for example, in the context of brain organoids, one can model the very earliest aspects of neural development in a platform that we call neuraloids that I will cover in a few minutes, or more complex synaptically active networks of neurons or neurons with different forms of glia that adopt fates that are specific for different parts of the brain. For example, in the context of Huntington's disease, cortical neurons or basal ganglia neurons that can be used to study the cellular impact of these mutations. The important component here is that this is a very versatile approach that can be applied to any indication and any human tissue type. The platform is mature, well-defined, can be scaled, and it leads to reproducible results. How we're using at Rumi the incorporation of organoids is really front and center of our drug discovery campaigns. The beginning is obviously modeling diseases by generating either isogenic lines or working with patient-derived stem cells to model cellular aspects of the disease or cell-cell interaction alterations in the disease. But these cells can be engineered to interrogate the output of genome-wide association studies that have revealed, in many cases, up to 100 different loci in the genome as being able to, in humans, modify some aspects of the progression of the disease. The biggest challenge in this area is to find quantitative phenotypic impact of these mutations to prioritize target selection for therapeutic discovery. We can also apply human tissues for predictive organ toxicology. We're using this in the kidney and in the CNS. Other people are using this in the context of kidney, neuromuscular junction, or muscle toxicological findings. We have adapted this technology for high-content screening, phenotypic screening that is compatible with chemogenomics or CRISPR-based genomic screening, enable whole genome screening for target identification and validation. And finally, on the right, we use it for the identification of on-target biomarkers. Once you have molecules that can tweak a particular biological process, and establishing the relationship between the drug exposure and proximal on-target biomarkers that can be further developed into pharmacodynamic biomarkers later on in development. At Rumi, we're using the organoids as well, connected in the drug discovery process, and I'll give you an example in Huntington's disease. Okay. This slide is meant to show the incredible beauty of these three-dimensional organoid systems. You are seeing here a picture on the right of an organoid that we call a neuraloid, which is the earliest stages of formation of the developing nervous system, with the four fates that are normally generated in the embryo very, very early on during gastrulation, where the emerging neural tissue gets generated and patterned. There are four significant cell types. You can see that the neuraloid mimics the three-dimensional structure in the human embryo, and it really enables, because of its configuration, enables image analysis to understand an impact of a particular mutation, particularly studying neurodevelopmental disorders in the context of the earliest fate specification in a system that is highly dynamic. The cells are dividing, the cells are moving, and becomes topographically organized in a very predictable manner. Something that is really almost miraculous is the fact that when you grow cells in the context, and this is a side view of this neuraloid, where you can see the very stereotypical three-dimensional organization of the emerging neural tissue that's marked here by PAC6, the neural crest that's marked by SOX10, and other tissues, including the skin, that becomes three-dimensionally dynamically organized into very predictable shapes and patterns that can be used with image analysis approaches to understand any perturbation in the system. The way micropatterns work is essentially a way to create with light polymerization that enables the control of shape and size of micropatterns. So you can see here in a 96-well plates, and you can see that there are about 30 different neuraloids or early neural development organoids that are essentially identical in shape and size. The physical constraints that are impacted with the micropattern trigger something miraculous in stem cells. You're seeding single dissociated cells. Those cells start to divide, but due to the physical constraints, they self-organize to create patterns, whether they're going to be patterns that resemble early stages of brain development, or whether they're going to generate all of these structures, for example, of a human mini kidney in this format. The robustness of this approach enables the analytical side, and the throughput enables a large-scale discovery campaigns. Okay, another component that is essential when you're trying to model different diseases or identify the activity of drugs or particular genes, in the case of GWAS, is to be able to have a very quantitative way of separating a disease state from a wild type unaltered state. The ability to generate large image databases, including in our case more than 1,000 microtissues per condition, enables an excellent separation between the impact of a particular mutation or permutation, and using image analysis to identify a potential ability of a therapeutic to restore abnormal cellular physiology. And this is the basis for a lot of the work that we do at Rumi. In terms of the internal pipeline, I'm going to tell you today about an example in Huntington's disease for a novel therapeutic target that we've identified, but we've applied broadly these neuraloid and other CNS organoid approaches to model the phenotypic consequences of genes associated with neurodevelopmental disorders or the autism spectrum disorders. And in the kidney, we've applied this to generate mini kidney tissues in the context of diabetic nephropathy or toxicological models that are associated with diabetic changes in the blood, as well as modeling monogenic forms of chronic kidney disease. Okay, I'm going to switch now into the Huntington's disease component. I know that there is another speaker that's going to talk probably about Huntington's disease. It's a disease that is very close to my heart. You can see a video of a patient that I met many years ago. It's a movement disorder characterized by involuntary movements. It affects about 1 person in 10,000 worldwide. I've worked extensively in developing therapeutics that are targeting the cause of the disease, the Huntington gene, and a lot of these programs are now in phase 2 or phase 3 in clinical development. I will tell you the appearance of novel therapeutics that are targeting the instability of the repeat that leads to the mutation that causes Huntington's disease. And also the fact that the biology, as we understand it today, is more complex than a simple toxic gain of function. And we have invoked that a loss of function of the normal protein is also contributing to the symptom progression in the disease. I think it's also of relevance to any neurodegenerative indication where the primary cause is being targeted, whether with small molecules or antisense oligonucleotides or siRNAs, that the therapeutics work best early. And there is evidence that I will tell you about that epigenetic changes are happening in the nuclei of neurons that are affected and degenerating the disease that render these primary disease-causing targeting therapeutics less effective as the disease progresses. And this is going to be a theme that I think is common to other neurological indications. Furthermore, Huntington is an essential gene from brain development, and therefore developmental systems can be very powerful systems to understand the primary pathological mechanisms that can be modeled using organoids. The Huntington gene is a very complex gene with 67 exons. There is a repeat in a polymorphic region in exon 1. It's a CAG trinucleotide repeat. The number of repeats is essentially what leads to the manifestation of the symptoms. The higher the number of repeats, the earlier the age of onset of neurological diagnosis. And in some cases, when people inherit a repeat that is greater than 60 CAG repeats, they develop the disease in the juvenile or pediatric ages. The youngest person I've seen with Huntington's disease is 3 years old, and he died soon after. So the impact of the mutation can clearly have a neurodevelopmental component to it, and therefore developmental systems like stem cell-derived organoids are ideally suited to answer basic and therapeutic questions about the pathological mechanisms that this mutation triggers. As I said before, the length of the CAG repeat is highly correlated with the age of onset, but it is well understood that some people born with the same number of repeats can experience the beginning of the clinical manifestation of the disease at very different ages, a span of 30 or 40 years. Therefore, this led to the identification of genetic factors that would affect the progression prior to the clinical diagnosis of the disease. These genes were shown in a large GWAS study to affect somatic instability of the CAG repeat in postmitotic cells, including neurons, and this is an area that is being actively pursued in the context of novel therapeutics. However, it is known that other genes have been identified that modulate the progression of the disease at the earliest stages that do not work through somatic expansion, and therefore there are multiple converging mechanisms that can modify the progression of the disease, even in the context of being born with a mutation that causes Huntington's disease. This is reflected here in the sense that the brain of HD patients is a mosaic. You are born, in this case, with a 40 repeat that should lead to adult onset HD, but some of the cells, when they've been sequenced post-mortem from individuals, we can see that some of the cells have large expansions, including in the hundreds. And this is thought to be a threshold that triggers the degeneration of those cells. So in the context of HD, there is a very long prodromic phase that lasts approximately 20 years. We can see that some of the earliest biomarkers, including MRI-based volumetric measurements or changes in cerebrospinal fluid or by PET, that the disease starts approximately 20 years prior to the diagnosis of the motor onset. And that these variants that have been identified through GWAS are critical modifiers of the age of progression to diagnosis. Okay. It is very important as well in the context of therapeutic development that the disease doesn't evolve everybody in the same way. And also, it doesn't always affect degeneration of the same circuits. And therefore, the clinical manifestations of the disease is highly variable in people. And it is related to the types of degeneration within networks within the basal ganglia that underlie those clinical manifestations of the disease. This is important because you need to try to stratify patients' populations for clinical trials, but also it's important to realize that there is broad spread degeneration within the cortical areas that needs to be accessible to potential therapeutics. And as we know, large molecules like ASOs or siRNAs don't get into the brain efficiently. In the context of current clinical development programs, we can see three major modalities: antisense oligonucleotides, gene therapy vectors that are directly administered into the basal ganglia in the context of HD, and a new set of oral drugs that affect the splicing of the Huntington gene and get everywhere in the body. And this is a summary of where they are in the context of clinical development. I think it's important just to highlight that multiple therapeutic agents are now in phase 2, being able to show statistically significant small improvements at a clinical level, not only in the motor domain, but also in the cognitive scales, in changes in volumetric MRI scans, as well as lowering the levels of neurofilament light protein, which is a good marker for neurotoxicity. So there's a lot of hope in the field that this is going to lead to an approval for the first time for Huntington's disease. Now, not all of the therapies are targeting all of the pathological mechanisms, and this is something to keep in mind. In the context of the Huntington gene, a recent discovery in the last few years has been that due to the expansion in the CAG repeat, one can generate only in the context of the mutation a truncated, very toxic form of the protein, and this protein is not being targeted by all of existing phase 2 and phase 3 therapeutic programs. Some of them lower it, some others only lowering or are only lowering the mutant version of the Huntington protein. It's also important.
Ignacio,
Yeah,
time
10
check.
minutes.
You have 10 minutes left,
Yeah.
20 slides to go.
Yeah, it's also important to realize that early treatment leads to a particular abrogation of most of the phenotypes in the preclinical species, but when these same therapies are applied mid-disease course, there is a refractory process at the level of nuclear pathology that seems to indicate that epigenetic changes are playing a role in making these therapeutics or making the disease refractory to Huntington lowering or somatic instability modulation therapeutic development. It is important as well that this has been associated with chromatin alterations in HD that therefore lay the course for novel therapeutic development to try to overcome the barriers to Huntington lowering therapies. Okay. I'm going to tell you a little bit now about the work that we did at Rumi. We essentially generated a set of isogenic cell lines with different lengths of the CAG repeats. We applied this to a Neuraloid platform technology, and they revealed CAG length-dependent deficits in the very earliest stages of the development of the nervous system. This can be exemplified here, but the expansion in the neural component in the Neuraloids marked by PAC6. And this suggested that the mutation was leading to quantitative changes that could be modeled very efficiently in the neuraloid, something that was not previously identified up until this point. We were able to then develop quantitative measures and conduct a chemogenetic screen that revealed the discovery of bromodomain inhibitors, which are epigenetic modulators. And we conducted a set of studies trying to identify the mechanism of action by which these non-selective inhibitors were able to rescue the mutant Huntington deficits. We took an siRNA approach in vivo into a mouse model of HD, showing that this early human organoid-based discovery could be translated in the context of the adult brain in a well-validated model of Huntington's disease. It's just an image that I wanted to show you. Essentially, what this image is saying is the phenotypes that were identified in an HD context compared to the wild type, and the fact that some small molecules that are targeting bromodomain inhibitors can completely restore the phenotypes back to the wild type context. This set of studies were validated using conventional image analysis or a neural network-based analysis of multiple features. And you can see here that the treatment of a BRD9 and 7 dual inhibitor, BI7273, was completely able to restore the phenotypes in the organoids back to a wild-type context. We extended these observations by trying to knock down individual genes, BRD9 or BRD7, and were able to converge that BRD9 was the gene that was mediating the effects of the small molecule inhibitors. And therefore, either suppression or inhibition of the enzymatic activity of BRD9 were valid therapeutic approaches for these phenotypes that we uncovered. We were able to extend, that this is one of the main challenges in the organoid field, which is to try to make sure that the activities that you discover are not just limited to the initial phenotypes that you're screening with organoids, but rather you can extend it in different cellular contexts, most importantly in the context of the in vivo brain. And in the work that we've done, we can see that BRD9 inhibition is able to also restore degeneration in vitro in other types of HD cell cultures that are conventionally used. In the context of the in vivo evaluation, because we knew that silencing or inhibiting the active site phenocopied the same rescue effects, we took an siRNA in vivo in an HD model and assessed its impact on disease progression. What we can see here is that the therapy is well tolerated in the context of the mouse. There were significant improvements in some locomotor endpoints assessed in this very aggressive model of HD. We were able to show that it decreased nuclear pathology or aggregated Huntington pathology in the nucleus very significantly with an siRNA that lowered BRD9 by 50%. And this was accompanied by a preservation of volumetric changes in the brain of these animals, as well as evidence of neuropathological rescue as measured by cell death or DNA damage response that is typically seen in this animal model of HD. This led us to begin to focus on BRD9 as a mechanism of action that we would like to target therapeutically for HD at an epigenetic level. And it's become obvious since then that Huntington itself forms a direct complex with the SWIFT SNF chromatin complex that BRD9 is a part of. These are complexes that are implicated in DNA repair and epigenomic remodeling in the context of brain signaling, and is a very novel area of active investigation for CNS disorders. We took advantage of the work that had been done in oncology to use a structure-based design to try to make and optimize BRD9 inhibitors for a CNS indication. The majority of existing molecules did not have brain exposure that was sufficient and pharmaceutical properties that were sufficient for advancing a therapeutic for the CNS. We were able, this is a program in lead identification at the moment, we were able to establish cellular assays on target activity using nanobread and showed a very good correlation between the activity of novel molecules for the target and the ability to rescue the deficits in the neuraloids. This is part of the reason we're using organoids in the context of drug discovery. This is a couple of examples here with some of the molecules that we've optimized in terms of potency and in terms of eliminated off-target toxicity that could be seen due to off-target activities of the molecules. And therefore, we're also using the organoid technology to set up the ability to distinguish programs that have very significant rescue effects on the organoids in an HD context with no toxicity. The way the program is right now, we've shown that we have functional rescue in the neuraloids. We have very good selectivity of the molecules for BRD9 and 7, and we're beginning to optimize the pharmaceutical properties in the context of the CNS. We have 3 different series, and one of them is brain permeable. The way we are trying to optimize the molecules right now is mostly at the level of brain permeability in terms of PGP efflux and also in terms of improving metabolic stability. And with that, I would like to conclude that we've identified BRD9 through a phenotypic screen using HD organoids. The technology is mature enough to support all aspects of drug discovery. The organoids reveal cellular phenotypes that are primarily connected to the first function that you can identify for disease-causing genes that are not typically detected with conventional systems. And for the longest time, we didn't have a primary function of the Huntington gene in a way that could inform drug discovery campaigns. We've identified in the context of BRD9 3 independent series that are in the lead identification stage, and I hope that maybe next year I can come back and tell you about our advancements in developing a epigenetic mechanism in the context of Huntington's disease. And with that, I would like to thank you for your attention, and thanks to the organizers again.
Thank you, Nacho, for excellent presentation. So let's see any questions from the audience. Okay, I see questions start to pour in. Maybe we start from this one. How does the heterogeneity with organoids array affect interpretation of the results right off bat?
Yeah, well, that's an important question, and it really depends on the biological questions and the type of organoids. The more complex the organoids, the harder it is. From my perspective, we're not using organoids in the conventional sense. I'm sure you will see in the next few seminars how other people are applying organoid technology that tends to be much larger, very heterogeneous in nature. The ability to use this geometrical confinement, even though if it has some limitations in the context of aging the organoids, it ensures reproducibility of the cellular composition and the patterning in a way that really enables image analysis to do its job. So in our case, our micropattern organoids are highly, highly standardized and quantitative. And that's one of the differences that our technology brings compared to conventional organoid systems that you will hear about in a second.
Okay, so maybe one more question before our co-host have a lot of questions. So how you really control the size of your microorganoids on this? What kind of secret sauce you put into the gels?
Yeah, well, I can't tell you about the secret sauce, obviously, that's proprietary, but this discovery was made in an academic context, and the idea was to try to understand how geometrical confinement and essentially mechanical cues induce the self-organization of tissues that happens during embryogenesis, and it's essential to the proper patterning of any tissue that would including conventional organoids. What that does is that by using light, you can create any pattern, any shape, any size you want. So I just showed you in the context of the neuraloids. In other contexts, for example, in the context of the more mature combinations of neurons and glia, the size of those micropatterns is much larger, and you need to optimize them depending on the system and depending on the questions. So it's highly malleable to the organoid or tissue system you want to study.
Depends on the stage of the development. It depends on cell type.
Right. And again, you have to play with the composition of the cells and the ability to quantitate the phenotypes that you can do. So it's essentially empirically determined. But because you're using light, you're not limited in any way. So basically, the light induces the polymerization of a matrix, but you can put masks that are light controlled, and therefore the cells grow in the areas where the gel is not polymerized. And because you're using light, you can create a string, you can create a triangle, you can create a home, you can create anything you want. And we can demonstrate with the technology what's more optimal for a particular application.
I see. The size of the polymerization is controlled by the light. Okay, so
Yes, so there's this, Dr. Ignacio, uh, this wonderful work, and I really enjoyed your work on the neuraloid development and how, it mimics brain development, and you can use those systems for studying and drug development for these developmental disorders like HD and autism spectrum disorders. So one of the questions is, do the kidney organoids also form structures found in the regions of the kidney? Like, do you see tubules? And in the lung organoids, do you see alveoli? Like,
Yeah. So if somebody's interested, they can contact me. I could give another talk about the kidney, but this is a CNS symposium. But yes,
right,
we
yeah.
have mini kidney organoids, essentially reproducible, that have all of the classical specifications that one sees, including endothelial vasculature. And we've conducted quite a bit of work in terms of transport assays and normal, how would I say, normal injury response to in the context of diabetic nephropathy or in the context of polycystic kidney disease. So the answer is yes to all those questions.
Wonderful. Yeah. Yeah.
So maybe one more question, Nacho. So you use both the embryonic stem cell as well as the iPSC derived.
Yes.
Oh,
The
correct.
one of the biggest challenges in the organoid in the stem cell field is the heterogeneity of how iPSCs were initially derived. So what we typically do is we move away from using iPSCs initially. What we do is we generate isogenic lines in lines that we have the rights to use that are highly compatible with our technology. We identify phenotypes and then we test those phenotypes in the micropatterns with patient-derived iPSCs. When you have 4 or 5 different iPSCs, you have isogenic lines that you can now correct the phenotypes. For example, I didn't show it, but you can lower the expression of mutant Huntington, or you can correct an isogenic mutation back to a wild type and see that the micropattern organoid deficits are restored. Then you have confidence that that phenotype is robust and applicable across different patients. That's the point in which we trigger the drug discovery efforts.
Excellent, excellent. Of course, there will be more questions, so we can discuss more in the panel discussion. Thank you.
Thank you.
So much.